next week that we use to study DNA in the lab, and we'll move on to carbohydrates and eventually into actual pathways, signal transduction, metabolism, things like that. All right, so you remember from chapter 1, right? DNA is biopolymer. It's one of those macromolecules that we talked about. And here we see the overall structure of DNA. Right? So it consists of a backbone made up of a deoxyribonucleic sugar linked to a phosphate, and that phosphate is in a phosphodiester bond with the next sugar. So we have sugar, phosphate, on and on and on. Remember, phosphates have a negative charge on them. So the backbone here repeats and is always negatively charged. The structure of the backbone never changes. On each sugar, in addition to being connected to the phosphate, in two places, right? One at the 5' end and one at the 3' end, as you'll see in just a minute. At the 1' position, we also have a connection to the nitrogenous base. Right? We have 5 possible nitrogenous bases. 4 for 3 of them are common between DNA and RNA. One of them, obviously, uracil and thymine, vary between RNA and DNA. We can break down our nitrogenous bases into 2 categories: our purines and our pyrimidines. The purines are bicyclic, the pyrimidines are monocyclic. That's an easy way to remember it. The purines consist of adenine and guanine. We find adenine and guanine in both DNA and RNA. These don't change. And if you notice here, the numbering of the carbons, they don't have a prime. It's only when we start talking about a nucleoside or a nucleotide together that we put the prime on the carbon numbers here, right? So notice they're nitrogenous because they have many nitrogens. Interestingly, right, also notice that they have many hydrogen bond donors and acceptors. Here we have a hydrogen bond donor, a donor, and an acceptor. Here we have a donor, acceptor, and nothing, okay? So these sides, the 6-membered rings of our purines, are we're actually going to be forming our Watson-Crick base pairs with their complementary bases on the opposite strands, as you'll see in just a minute here. But what determines which ones these pair with comes down to the pattern of acceptors and donors, right? So we have, again, acceptor and donor, or rather the other way, a donor and acceptor, and here we have an acceptor, a donor, and a donor, right? So this pairs with our pyrimidines, right? We always get a purine pairing with a pyrimidine on the opposite strand. Our pyrimidines are cytosine, uracil, and thymine. Cytosine, again, we find in both DNA and RNA, but uracil and thymine are unique. Uracil we only find in RNA, and thymine we only find in DNA, okay? Now, we'll revisit this again later in the semester, but what's the difference between uracil and thymine? The thymine just has this methyl group here, all right? Now, why is this important? Well, as it turns out, right, cytosine can be oxidatively deaminated spontaneously. And what that means is that this amino group on carbon number 4 can spontaneously turn into a keto group. And what happens if we turn to cytosine, we remove the amino group and we turn it into a keto group? What is that? What is that structure? It's just uracil, right? So why is it important that in DNA we use thymine versus uracil? Why did evolution select for this difference between the 2 nucleotide biopolymers? Well, this oxidative deamination isn't rare, right? In fact, it happens thousands of times a day. So if we had a cytosine which spontaneously deaminated to form a uracil, now your cells, the nucleus, is supposed to be there, or it used to be a cytosine. So, when it synthesizes the deoxynucleotides, which is DNA synthesis, one of the steps is to take deoxyuracil monophosphate, okay, and convert it into thymine monophosphate, right? Just adds a methyl group. So what happens now? This cytosine were to be deaminated and form a uracil. Well, now the cell can say with certainty, okay, if I see a uracil in DNA, I know for a fact that that used to be a cytosine. Right? You can convert it back to preserving the sequence of DNA. And of course, what is the sequence of DNA telling us? Well, it's telling us what the instructions needs to live. So this helps to protect those instructions from a chemical reaction that happens naturally, right? So again, this cytosine plus H2O will yield uracil in a non-negligible at a non-negligible rate.
Is purine like just a category, or is it also a specific molecule?
No, it's just a category, right?
So then what is like that molecule pure? It's just generic?
No. I see what you're saying. No. This is pyrimidine, okay? All of the pyrimidine bases are based on pyrimidine.
But pyrimidine itself doesn't exist?
I mean, it exists in the universe, it is a thing, but not in DNA or RNA, right? So all of our purine bases are based on purine, okay? So notice here we just for adenine, we just aminated here. For guanine, we've oxidized here, and so on and so on. So yes, purine and pyrimidine do exist, and these are modified to form adenine guanine, or in the case of pyrimidine, cytosine or something, right? But neither of these exist in DNA or RNA. Yet they can be synthesized, and they are real molecules, but not in a biologically relevant sense. Good question. All right. So now, what is a nucleoside? Right. So we have our bases. A base plus either a ribose or deoxyribose sugar, right, forms what we call a nucleoside, not to be confused with a nucleotide. Right. So a nucleoside is just the base plus the sugar. For now, just memorize the structure of ribose. Okay. So this is, unfortunately, we haven't talked about carbohydrates yet, so just have to memorize the structure of ribose because I told you to. Right. But once we get into chapter 11 and start talking about carbohydrates, you'll see why the structure of ribose is this way, right? So all sugars, carbohydrates end in OSE, O-S-E. Ribose is simply that. It's just a sugar, right? It's a specific sugar. It's got 5 carbons. It's what we call an aldose, right? And if we notice here, we are numbering the carbons, right? 1', 2', 3', 4', 5'. We add the prime to distinguish them from the carbons of the nitrogenous bases. So the nitrogenous base carbons, or atoms, not necessarily carbon, have regular numbers, while the carbons, in this case, on the ribose or deoxyribose sugar have prime numbers. They have the same numbering, right? The only difference between ribose and deoxyribose is the missing hydroxyl on the 2' carbon. That's the only difference. So deoxyribose is more accurately described as 2' deoxyribose. We can have other deoxyriboses or dideoxyriboses, as we'll see in the next chapter when we talk about DNA sequencing. But ribose we find in RNA, that's the sugar component of the backbone, while deoxyribose we find in DNA, that's the sugar component of the DNA backbone. The 1' carbon is always connected to a nitrogen on the nitrogenous base. And it's the identity of this nitrogenous base that gives the nucleoside its name. It's linked via what we call a beta glycosidic linkage. A glycosidic linkage is simply a linkage between the anomeric carbon of a sugar, which we'll talk about in chapter 11, but in this case, that's just the 1' carbon, right? So it's a link between the anomeric carbon of a cyclic sugar and something else, right? Usually an oxygen. But in this case, it's a nitrogen, so we call it an N-glycosidic linkage. Beta in this case, because if you look at the stereochemistry of the linkage between the base and carbon 1', this base is on the same side of the sugar as carbon number 5, or 5'. So if you imagine that this is flat on the page, carbon 5' is coming out towards you, and so is the base, right? So if they're on the same side, we call that a beta glycosidic linkage. In this case, specifically a beta N glycosidic linkage. If it were on the opposite side, we would call it an alpha glycosidic linkage. Now, in the context of a nucleoside, we don't necessarily use alpha and beta, we use syn and anti, right? So syn means that they're on the same side, and anti means that they'd be on the opposite side. All nucleosides that make up RNA and DNA in a biologically relevant sense are in the syn configuration, right? So they look like this. They have the base and carbon 5' on the same side as, of the plane of the sugar. Now, a nucleotide is simply a nucleoside plus 1 or more phosphates, right? So it's a sugar, a base, and 1 or more phosphates. So what you see on the left here is ATP, right? So this is the ATP that we find in the cells that we all talked about in Gen Bio probably, all right? Now specifically, it's 5' ATP. Now, the way that we name, we'll come back to this slide in just a minute, the way that we name our nucleosides and nucleotides, okay, are adenosine, right? So they end in sine for the purines, and they end in dene for the pyrimidines, right? Pyrimidine, they end in dene, right? So that's easy to remember. Adenosine and guanosine are just the sugar plus the base, right? Of course, if it's a deoxy, we just put deoxy in front of it. a nucleotide, which includes the base, the sugar, and one or more phosphates, are called adenylate, guanylate, acetylate, and amylate, right? So they all end in late. So there's a little trick to remember. Now, we very rarely refer to nucleotides by their correct name, right? So we wouldn't refer to ATP necessarily as adenylate. The reason is, right, a nucleotide is referring to a nucleoside in one or more phosphates. Two things are missing here. What's missing is A, where are those phosphates, right? Which carbon are they on? And B, how many phosphates are there? So in order to remove this ambiguousness, okay, well, we put the carbon that the phosphates are attached to, and what we use is the nucleoside name plus mono, di, or triphosphate. So in this case, 5' ATP is 5' adenosine, which is the nucleoside name, triphosphate. So this specifies that there are 3 phosphates on an adenosine, and that those phosphates are on the 5' carbon. Now, usually this 5' is omitted. When the 5' is omitted, it's assumed, okay? We can also have things like 3' deoxy GMP, right? So what are we looking at here? This would be guanosine monophosphate on the 3' carbon, right? So this would be guanylate, right? But this is got the same name as GTP, which is, or 5' GTP, which would be guanylate or guanosine triphosphate. So we have 3 phosphates on the 5' carbon. There's no way to distinguish them. So usually, again, the way that we describe or name nucleotides is by using the nucleoside name, specifying how many phosphates there are and on which carbon they're attached. Again, it's not specified that 5' is assumed or implied. All right, now let's talk about the primary structure of DNA. Right, so DNA, like proteins, can have primary and secondary structure. Right, so the primary structure of DNA is similar to what we saw with proteins, is simply the sequence of bases. Right, so the backbone is always going to be the same. It's always going to be ribose phosphate, or deoxyribose phosphate, deoxyribose, so on and so on. Similar to what we saw with proteins that have a repeating backbone of N, C alpha carbonyl, N C alpha carbonyl, N C alpha carbonyl. That backbone is always the same in proteins. The backbone is also always the same in DNA, right? And we can kind of draw DNA in a shorthand, which is the stick and pea model. And in this case, right, I really hate the way the book does this because they kind of messed up here. But what we do is we draw a vertical line. The vertical line represents our sugar, either ribose or deoxyribose. Okay, the 1' carbon is at the top, the 5' carbon is at the bottom, 3' is in the middle, 2' is in between 1 and 3, and 4' is in between 3 and 5, right? So we have 1', 2', 3', 4', 5', right? So why I don't like the way the book does this, and I don't know why they haven't fixed this yet over like 3 editions so far, is that this 3' here on this deoxyribose really should be right in the middle, right? That's where the 3' is. But it's not. They've drawn it up here, so don't get confused here. But the 3' carbon of the first sugar is connected to a phosphate, like we see, I guess we haven't seen yet. We'll see in a minute, right? So the 3' carbon of one sugar is connected to a phosphate. That phosphate is connected to the 5' carbon of the next sugar. The 3' carbon of that sugar should be connected to a phosphate, which is connected to the 5' carbon, and so on and so on and so on, right? So it's a 3' to 5' linkage, meaning that on this end of a DNA strand, we always have a 5' connected to a phosphate, which is free, so we call this the 5' end, or the 5' phosphate. And on this end, right, we always have a 3' free 3' end, or a 3' hydroxyl, right? So again, like we saw in chapter 1, DNA has a polarity. We have a 5' end and a 3' end. It's not symmetric. Now, this isn't very commonly used, but it is good to be able to draw this just for your own sake, okay? But if we think about it, right, all we need to know are what the base is at carbon 1' on the first, carbon 1' on the second ribose, and carbon 1' on the third ribose, right? So in this case here, we just have A, C, G. Well, A is adenine, C is cytosine, and G is guanine, right? So we know the structure of adenine, we know the structure of cytosine, we know the structure of guanine. We also know that they're connected to their respective sugars always in the same way. And we know that those sugars are connected together to form the backbone always in the same way. So from 3 letters, A, C, G, you should be able to draw an entire, in this case, 3 DNA or RNA polymer. Now DNA can adopt a secondary structure, right? So we have the primary structure, the sequence of the DNA. But of course, we all know that DNA is double helical. This is the secondary structure of DNA, right? This is, of course, solved in 1953 by Watson, Crick, and Franklin. And of course, this is kind of the classical picture. This is the illustration that actually appeared in that 1953 paper. Okay, I'm not going to get into how they analyze the X-ray diffraction pattern, but this was discovered using a method called X-ray diffraction. Basically, they took a bunch of DNA, they crystallized it, right? And what is a crystal? But just a bunch of identical molecules stacked on top of each other, like salt, right? So it all stacks on top of each other. Weird thing about crystals is that because they're all stacked together and they're all identical, right? They all have the same orientation, so they will diffract light exactly the same, right? And we can measure that. We can essentially just shine an X-ray beam at a crystal of, in this case, DNA, and it will diffract that X-ray light. We can put an X-ray film on the other side, and this is the image that we can capture. Now, using some fancy math that frankly I don't know, right? We can convert our X-ray diffraction pattern into a 3-dimensional structure. In fact, this is how the structure of many proteins has been solved. Right? A crystal structure, or a crystal of protein is grown, and they shine a beam of X-ray light at it, they measure the diffraction pattern, and they can use Fourier transforms and things like that to determine what structure needs to exist in order to produce this diffraction pattern. In this case, that is a double helix. Right? Now if you notice, this double helix isn't like the double helix that you see in cartoons where we have 2 evenly spaced DNA strands. In fact, it's asymmetric. We have what we call a minor groove and a major groove, right? One of these sides of the double helix is smaller than the other side of the double helix, right? So it's not symmetrical. And we can see that better over here, okay? We have a major groove and a minor groove. Now, interestingly, right, here we have our base pairs. We saw this again in chapter 1. We have an A base paired with a T, 2 hydrogen bonds, and a G base paired with a C, 3 hydrogen bonds. And right? Think about it. Here's our base pair, okay? Well, one side of this must be the major groove, and the other side of that base pair must be the minor groove. So we can see that here. Between an AT base pair, we have one side that constitutes the major groove and one side that constitutes the minor groove. And the same is true for a GC base pair. One side is major, one side is minor, right? Why does this matter? Well, if we see, of course, we have the hydrogen bonds that are forming the Watson-Crick base pairs, holding these 2 together, right, or ensuring their specificity. But those aren't the only hydrogen bond donors and acceptors that exist on these nitrogenous bases. For example, here in an AT base pair, we have a hydrogen bond acceptor here, a hydrogen bond donor here, another hydrogen bond acceptor over here. Okay, so if we have something that can recognize that pattern of hydrogen bond donors and acceptors, well, now we have something that can recognize an AT base pair, right? And we can use the same principle to say, we want to recognize a TA base pair, right? So if we swap around their strands, the pattern would change. And the same is true for a GC base pair. Here we have an acceptor, donor. Here we have an acceptor, donor, acceptor. Right? So there's a difference in the pattern, and there's a difference in the pattern when the base pairs are replaced. So instead of an AT, if we had TA, that can also be distinguished. In fact, any DNA binding protein that has to recognize a specific DNA sequence, this is how that's achieved, right? By making their active site complementary or able to form hydrogen bonds with the major groove. Not the minor groove though. The minor groove, of course, is much smaller, so there's less room for a protein or enzyme to fit. Additionally, there's less information available here, right? Here, notice we have an acceptor, while here we have acceptor. The only difference is that we have one donor here, okay? So in addition to being smaller in size physically, there's less information there. So DNA recognizing enzymes typically look at the major groove. This is how DNA specificity is achieved, right? So you can imagine you have an active site that recognizes an AT base pair, okay? by forming hydrogen bonds, but close by, in that same active site, there's another series of residues that might recognize the pattern on a GC base pair. And further on, there might be one that recognizes a CG base pair, and so on and so on, right? So by having different patterns of residues in the active site of a hypothetical DNA binding protein, now you can start to put together a sequence, right? By matching up the pattern of residues that can form hydrogen bonds with the major groove of our DNA double helix. Now, if we look at the actual structure of the double helix, of course, again, we have the minor groove and the major groove, but we have a rise of 3.4 angstroms per base, okay? We have a pitch of 34 angstroms per 360-degree turn. And this gives us a pitch of 10.4 bases per turn, right? Similar to how we saw we have 3.6 base residues per turn in the alpha helix for the secondary structure of a protein. Here we have 10.4 bases per turn, or 360 degrees. Now, for this class, when we start using these for calculations later on in the semester, we're just going to round this down to 10, right? So we're going to round this down to 10 base pairs per turn. You're not going to need this now, right? So we're not going to talk about this in chapter 8 here, but we are going to round this down. But you should know that the base separation, the rise is 3.4 angstroms per base, okay, and that the pitch is 34 angstroms per turn, right? Now notice again that each of these base pairs are more or less perpendicular to the axis of the helix, and they're more or less parallel to each other. Does anybody remember what that phenomenon is called? That these base pairs are parallel to each other.
Base stacking?
Yeah, right, base stacking. And what weak force mediates the base stacking interactions? Right, van der Waals interactions, okay? So these remain perpendicular to the axis and parallel to each other, mediated all by van der Waals interactions. Now, if we look from the top down, right, we see here we have some purine-pyrimidine base pair here, right, and here and here. So we have a twist of 36 degrees per base pair, right? Of course, that makes sense. If it's 10 base pairs per turn and there's 360 degrees, that means that each one needs to be twisted 36 degrees relative to the one above and below it, right? 20 angstroms wide, okay? And this is what we call bDNA, right? bDNA is the hydrated common form of DNA. This is the DNA you find in all of your cells. We're going to see another form of DNA shortly that's called aDNA. Now this you don't find in a biologically relevant context really, because it's DNA devoid of water. Now if you go on RCSB, the protein data bank that's got all the structures for basically any protein or molecule for which the structure has been solved, you can find structures of DNA, right, that have been solved either by X-ray crystallography or NMR or other methods, okay? All of those, with the exception of the NMR ones, are going to be aDNA, not bDNA. Reason being, in order to form a crystal, what do you need? You need only that particular molecule, right? Anything else, any other impurities that get into your crystal will damage the structure of the crystal. One of those impurities being water. So in order to get the structure for DNA in crystal form, you need to exclude all the water, which of course makes it adopt the A form. But the B form is what we find in most of our cells, right? In all of our cells actually, except for erythrocytes. But we'll see what aDNA looks like in just a minute. Now the base compositions can be experimentally determined, right? And this led to an interesting observation, right? So Erwin Chargaff, it's called, well, the rule is named after him, Chargaff's rule. What he noticed was that, for example, if we look at the base composition of human DNA, he found that the ratio of A to T is always exactly the same as the ratio of G to C. Or rather, the ratio of A to T is always 1, and the ratio of G to C is always 1, meaning that they're equal to each other, right? But what he observed was that the ratio of A to G or T to C had no pattern, right? It could be 1.56, 1.05, could be less than 1.7. So there's no relationship here. But what he noticed, again, was that the ratio of A to T is almost always exactly 1, and the ratio of G to C is almost always exactly 1. Now the variation here, right, could be due to experimental error, okay? And what does this show, right? Well, this shows that however much A we have to have a T, right, in order to have that ratio be 1. And however much G we have, we have to have a C. So this, notice, was in the 1940s. Right? So this is before the structure of DNA was solved. So we didn't know that there were these base pairs that came together. This was used by Watson, Crick, and Franklin to refine their model. They knew there must be some relationship, some interaction between A's and T's and between G's and C's such that Chargaff's rules always hold true no matter which organism we look at. Right? The ratio of A to T and G to C should always be about 1. Right? So how could there be this relationship? Well, this pattern here let them know that there was some sort of helix being formed. Right? There's some sort of helix being formed. But the pattern of spots here, right, told them A, the periodicity, and also that there were 2 helices. So how could these two be interacting then? The A to T and G and C, how could they be interacting? Well, we say that these helices themselves interact, and that wherever we have an A on one helix, we have a T on the other helix, well, that would satisfy Chargaff's rules here. Right, so it's an example of, even though we might not have the full picture at any given point, right, now we know a lot about DNA, but there's a lot of things that we still don't know about. For example, the sugar code, right? So how sugars are linked together and used to modify proteins to signify different things, right? So a whole different field of biology, right? Small increments, right? Small increments in understanding can help to lead to more a complete picture, right? So the structure of DNA couldn't have been solved without this observation made by Chargaff before the structure of DNA was known. All right. So again, here we have the base pairs. So again, the base pairs are stacked on top of each other, mediated by van der Waals interactions. Remember, there's a van der Waals contact distance for any given atom. If we move them too close, the energy increases. If we try to move them apart, well, that requires an input of energy. So the stateless form, or the stateless state of this system can have 2 base pairs just stacked parallel on top of each other, and the van der Waals interactions keep them, or the van der Waals forces keep them from moving apart or too close together. If we look at the GC base pair, right, this is where we said before, okay, this side of the purine matches the pattern of hydrogen bond donors and acceptors on this side of the pyrimidine, right? So here's our pyrimidine, cysteine, and our purine, guanine. Here we have a hydrogen bond donor forming a hydrogen bond with the acceptor, the corresponding acceptor on cytosine, this acceptor with this, or this donor rather with this acceptor, and this acceptor with this donor. And this also led to, or helped lead to, the overall solving of the DNA structure, right? How did we come up with the idea that the DNA strands had to be anti-parallel? Remember, we talked about that in chapter 1, where one strand goes 5' to 3' in one direction, while the other one goes 5' to 3' in the other direction. Well, if we had this cytosine, right, on the opposite strand, remember these are on anti-parallel strands, if we made the other strand be parallel, right, well, what would happen now? We would have an acceptor next to an acceptor, so there would be no hydrogen bond. This would still work, but now we'd have a donor next to a donor, so that wouldn't work, right? So it was examining the structure of the bases themselves and trying to take into account Chargaff's rule. There should have been some interaction between a G and a C that finally helped to solve the structure of DNA, right? Now here we see the differences between A-DNA and B-DNA. Again, here the rise per base pair in B-DNA is 3.4 angstroms that we talked about before. A-DNA is more squat, right? So it's kind of shorter. That's got a rise per base pair of 2.3 angstroms. You don't have to memorize this. But if we see the structure of A-DNA, well, we can see that, yes, it is in fact squashed down. And when you squash it down, you spread it out, so the diameter here is 26 angstroms compared to 20 angstroms. But they're both right-handed, right? So DNA is a right-handed helix. A-DNA is also a right-handed helix. They both have their glycosidic bonds in the anti-configuration, right? Base pairs, well, the pitch right here is 11, while in B is 10, right? So we see that it's got different characteristics. A-DNA, again, is not something we encounter biologically. And even weirder is Z-DNA, right? So Z-DNA is long and skinny, and it's zigzaggy, right? So it is a left-handed helix, first of all, okay? It's taller, it's got a rise of 3.8 angstroms, It's narrower, it's got 18 extras, so it's tall and narrow, the opposite of A DNA. What's weird here is that it's got alternating anti and syn glycosidic bonds, right? So while in A DNA, they look exactly like they did on that slide before, okay? Let's see, not too far. Here, right, in RNA and DNA, all of the nucleotides look like this. In Z DNA, there's an alternating pattern, right, which gives it a zigzag shape. Now, the reason I don't even have a picture of it up on the screen is the significance or relevance of zDNA is still not entirely known. We know that it is a form of DNA that exists. It has been observed, right? But we don't know why it exists, when it exists, and for what purpose it exists, right? So I'm not really going to talk about it any further than that. Now, DNA specifically, and RNA, as we'll see, can adopt a tertiary structure as well. Okay? Now, for DNA, it's a little bit complicated because it's a split between eukaryotes and prokaryotes. Prokaryotes is easy because prokaryotic organisms, their DNA chromosome is arranged in a circle, right? So the 5' end, well, one end, the 5' end on this side and the 5' end on this side come together and form a phosphodiester bond so that we have a full circle. Now, circular DNA can do something very interesting. We'll see this again in chapter 28 or 29. They can form what we call supercoils, right? So here we have an example of what we would call relaxed DNA, right? Or relaxed circular DNA. So this would be maybe from an E. coli chromosome. And what we see is that topologically, all right, it lies flat, right? So here it's not, a perfect circle, but we see that the axis of the double helix, this is an electron micrograph, the axis of the double helix never crosses itself, right? So this is just a circle of DNA, right? We can go in there and kind of straighten it into a circle if we had tiny enough fingers. But by breaking open the circle, either under twisting it or over twisting it, right? So we twist it a little bit more and put them back together. Now we form a kink, right? And that kink has to be resolved, and the way that it resolves that is by twisting the entire structure, right? So it forms into a figure 8. Again, that's called the linking number of DNA. We'll talk again more about this in chapter 28, 29. But this whole structure here, for example, the relaxed form of the circular DNA is about 100 times larger than an E. coli cell, right? So the chromosome of a bacteria shouldn't actually be able to fit inside of a bacteria. It's just way too large. So how do they fit their entire genome, their entire chromosome into their cells? Well, through supercoiling, right? So here, notice this has been supercoiled many times. Here we have one crossing of the helix here, right? So the axis of the double helix crosses itself here, it crosses itself here, it crosses itself here, all over the place. And the effect of this is to reduce the size of the chromosome, the physical size, right? So this is a very mildly supercoiled DNA, right? Supercoiled because it's a coil made out of coils. So here we've got maybe 10 or so crossings of the axis of the double helix. But in reality, we have many thousands of crossings. So the linking number is actually quite high. Again, we'll see this in chapter 29. By doing this, again, we can shrink the effective size of DNA so that it can fit into the cell, right? Now, eukaryotes do this slightly differently because eukaryotes don't have circular chromosomes, right? Our chromosomes are linear; they have a beginning and an end, right? So how then can we do it? Because remember, how do we achieve supercoiling? By breaking open a circle, twisting it or untwisting it, and then gluing it back together, right? But let's try to do the same thing with a linear chromosome, where we can untwist it, we'll just twist back to what it's supposed to be, or we can over twist it, it'll just twist back to what it's supposed to be. So how do we supercoil linear DNA? Because we do have to supercoil our DNA. We have the same problem that bacteria have. Our DNA is simply just too long to fit inside of our cells. So we do the same thing with the help of accessory proteins called histones, right? It wraps around, or the DNA wraps around the histones. And when this happens, right, this effectively over-twists it and holds it that way. And we have many, tens of thousands of histones per chromosome, which help to, just like in this case, shrink the effective size so that it can fit in our cells. right? An analogy I've heard, I've never actually done the math, it's like trying to take a spaghetti the width of Africa and trying to fit it inside of a basketball, right? That's what histones allow us to do, whereas in bacteria, all they have to do is to supercoil their DNA, so not require any accessory proteins, with the exception of the enzymes necessary to actually achieve the supercoiling.
Wait, what do you mean the width of that?
At the widest one.
Oh, like going from one side to the other.
This way. This is good. Alright. That's not going to be on the exam. RNA can also adopt tertiary structures, right? So of course the main difference between RNA and DNA is that DNA is double-stranded and RNA is single-stranded, right? This limits the amount of tertiary structure that a DNA can adopt, right? Again, pretty much supercoiling or circling around histones is the only type of tertiary structure that a DNA molecule can adopt. RNA molecules are much more flexible though, literally, and in the types of structures that they can adopt, tertiary structures. Reason being, all right, that RNA molecules are able to form intramolecular helices, okay, which is very important. So let's look at this molecule here, right? Everyone thinks of RNA as just kind of messenger RNA. It's a temporary molecule, how you write notes for the ribosome to follow, right? But RNA molecules actually have much more interesting purposes than just serving as messenger RNA, right? So let's examine first how we can form an intramolecular helix or intramolecular base pairing. Well, here we have an RNA molecule, right? And we go from 5', we'll follow this all the way around to 3', okay? Now, because the bases in RNA and DNA are the same, in theory, they should be able to base pair the same, right? So we have an A, we should be able to base pair with a T elsewhere in that RNA molecule. But remember, we have to have the two strands oriented in an anti-parallel manner in order for the pattern of hydrogen bond donors and acceptors to match between opposite bases, right? So let's see if that's the case. Well, here we'll start, right? So this is the 5' end of the U, and we'll go up, and this is the 3' end of the C up here. So we have 5' to 3' in this direction. Now we'll make this hairpin turn, okay? And what do we have here? Well, this is the 5' end of G, and going back down, this is the 3' end of A. So we have 5' and 3' down here, and we have 3' and 5' up here. So these two segments of the same RNA molecule locally are now anti-parallel to each other. So the pattern of hydrogen bond donors and acceptors between this C and this G will match, and the same between this U and this A, right? Notice this no T here, it's a U, right? So we can get intramolecular base pairing. So this is an example of a structure in an RNA molecule that we call a hairpin. Right? It looks like a hairpin. But we can adopt way more complicated structures, right? So here's an RNA molecule, and we see that here we get intramolecular base pairing down here. Here we have a hairpin. Here we have another hairpin down here. All right, A with U, G with C, and so on and so on. Additionally, right, we can form non-Watson-Crick hydrogen bonds, right? So the Watson-Crick hydrogen bonds are going to form between complementary bases on the same RNA molecule in this case, okay? But remember, we have other hydrogen bond donors and acceptors that aren't participating right now in Watson-Crick base pairing, okay? So that's what we see over here. We have a non-Watson-Crick base pair between this Watson-Crick base pair, GC, and this A over here. So we have the G and C over here with its 3 hydrogen bonds in black. These are the normal Watson-Crick hydrogen bonds that we saw a couple of slides ago. But this adenine over here, right, which notice is not in a Watson-Crick base pair because it's opposite a G in this case, has all of its hydrogen bond donors, acceptors, available to form interactions, and indeed it does. It forms hydrogen bonds with this on the guanine, with this on the guanine, with this on the cytosine, and this on the cytosine. So these hydrogen bond interactions between this and this help to hold this region together, right? Same could be said for any of the other ones, right? So here we might have non-Watson-Crick hydrogen bonds holding these two regions together and all over. The effect of this is that these intramolecular base pairs, Watson-Crick base pairs, help to define the structure, and these non-Watson-Crick base pairs help to stabilize the tertiary structure, okay? And what is this starting to look like? I don't need a specific thing, right? But in general, what is this starting to look like? Huh? You could say it's a supercoil, right? But more generally, it's starting to look like a protein, right? It's got a defined structure, okay? In fact, right, well, not many, but a lot of RNA molecules form these types of structures, okay? And they can act as enzymes, and we call those specifically ribozymes. For example, the ribosome. The ribosome is made up mainly of RNA. By weight, right, it's something like 70 or 80% RNA by mass, right? What does that mean? Well, once we start talking about protein synthesis in chapter 30, we'll see that the actual enzymatic activity of the ribosome, in other words, the actual peptide bond formation and the recognition of the messenger RNA, and all this is mediated not by protein, it's mediated by RNA, specifically the 23S ribosomal RNA, which acts as a ribozyme. And how is it able to act as a ribozyme? Well, it forms a lot of hairpins, a lot of bulges like you see here, okay, by intramolecular Watson-Crick base pairs, and the overall structure is stabilized by non-Watson-Crick base pairs so that it can have a defined structure. Remember, the structure of anything defines what it does. Right? Now, messenger RNA tends to not have a structure because that's not doing anything except being read by the ribosome. But other molecules, or other RNA molecules that have other functions, must have a specific structure, right? Go back over here for a second. Here, another example. We'll come back to this slide later on, but here's a transfer RNA, a tRNA. tRNAs, again, we'll revisit in chapter 30, but tRNAs must also have a defined structure so that they can be recognized by elongation factors and the ribosome. And these are held together by Watson-Crick base pairs in each of these three hairpins, and are stabilized by non-Watson-Crick interactions so that they can adopt the correct three-dimensional structure so that they can be recognized, right? So RNA molecule is actually quite versatile. There's a theory called the RNA world that the first really biological macromolecule to have existed was RNA. Reason being that RNA serves, or can serve, as both a repository of genetic information, right, by mimicking what DNA does, and it can also serve as an enzyme, right? So, in theory, the only polymer that a bacteria or any organism would need in order to replicate itself is RNA. You can have an RNA serving as an RNA polymerase, as a ribosome, as the chromosome itself, right? As we'll see in just a minute, viruses still use RNA as their chromosome, as their genetic storage medium.
So not all RNA will do intramolecular.
No, not all RNA will do this intermolecular base pairing and binding and all this, right? Only molecules that have a specific function. That's not to say that messenger RNA can't form intramolecular Watson-Crick base pairing, right? It might be able to if there are enough that happen to be complementary, but that's not the overall purpose of messenger RNA. And besides, it's going to be then recognized by the ribosome and pulled through the ribosome, so any intramolecular Watson-Crick base pairs that form a messenger RNA are going to be undone anyway, right? All these other molecules, or ribozymes, they do not have the specific sequence that they need to be recognized by the ribosome. So they're not going to be touched by the ribosome. Instead, are going to go off and do whatever these RNA molecules are supposed to do. All right, now, how is DNA replicated? All right, well, again, we'll get into the more nitty-gritty in chapter 29, but how DNA is replicated is suggested by its structure. Right? Once the structure of DNA was solved, actually, I forget if it was Watson or Crick speculated at the end in the discussion, right, that the structure of DNA suggests a way in which DNA can be replicated. We can see why. If we have two anti-parallel strands of DNA. And on this strand, if there's an A, the other strand must be a T at that position, and so on and so on. Well, now all of the information necessary to synthesize its complementary strand is encoded on the parent strand, right? So this was the theory put forward, right? We start with our parental DNA, and what we would do is to pull those two strands apart, okay? Each of those two strands now serves as a template for the synthesis of a new strand. And at the end, we should have two identical double helical DNAs, right? Because if we started with strand A and strand B, right? Well, if we take strand A away, we synthesize the complementary strand, what do we have? Well, we have a new B strand, right? But we have another B strand over here, and if we synthesize its complementary strand, that's just A. So we started with an A and B strand, and now we have two A and B strands. And that's what we see here, okay? The old strands, the parental strands, are in blue, while the new strands are red, right? So we have two identical hybrid first generation DNA molecules. Now each of these, of course, can serve as a template, right? So we pull these two strands apart, we can get this if we use the blue strand as a template, we get this if we use the red strand as a template, and the same over here, right? So notice we're going from 1 to 2 to 4. From here, how many are we going to be able to synthesize? In the next generation, how many will we get? 8. And then, right, so we get exponential amplification. Now this was just a theory at the time that the structure of DNA was discovered, right? But it was discovered actually what was considered to be one of the most elegant experiments in biochemistry, right? And this was the semi-conservative. Okay, so let's see how this works. So, in order to figure out, right, what was going on with the DNA as it's being synthesized, first thing that we need to do is to be able to distinguish, right, new DNA and old DNA. And how do we do that? Well, we know that DNA's got a lot of nitrogen, right? So what we did, well, not we, I was born yet, right? But what was done was that the bacteria they used for this experiment was grown up in a media where all of the nitrogen was replaced with N15, right, which is a heavy isotope of regular nitrogen, which is N14, right? So this is a heavy isotope of nitrogen, meaning that after many generations, all of the nitrogen in the bacteria, and by extension, all of the nitrogen in the DNA of that bacteria should have been N15, right? Now you can distinguish molecules that have identical chemical makeup, but differ only in, a proton by using density gradients, right? So here, we take our DNA solution, we put it in a density gradient, and we can separate out based on the density or weight of that molecule, right? In this case, because all of the DNA should have been identical, the only difference in mass should have been that proton between N14 and N15. Then you multiply that across, all of the thousands of nitrogen in a DNA double helix. Well, now you have something that can be easily distinguished in the density gradient, right? So the densest at the bottom would be DNA that's made up of entirely N15. So on both strands we have heavy nitrogen, followed by the lightest, which would be all N14, right? So we have two species that we could possibly have. We could have the heavy species, which is both strands being made up of 100% N15, and we have the light species, which is all of the strands, all of the nitrogens being made up of N14. And that's what we see here, okay? Here on this side, this is the bottom of the density gradient, right? And this corresponds with N15. Over here, we have slightly higher up in the density gradient. This is our N14 DNA, right? So after many generations, again, these bacteria grew up in a media containing only N15, so all of the nitrogen in the DNA should have been N15, and that's what we see at the top there. In generation 0, we have all of the DNA that was isolated from those bacteria moving to the N15 line, which is as expected. Now, we take those bacteria, we spin them down, we isolate them, we throw away the N15 media, right? We take those bacteria, we wash it to make sure there's no residual N15, and we take this bacteria and we put it into fresh media. And this fresh media contains not N15, it contains only N14. Okay, so we move the bacteria from an N15 environment into an N14 environment. And what happens? Well, now we let it grow for one generation, right? One exact generation. And what happens? Well, we isolate the DNA from there. Do we have any N15 DNA? Do we have any N14 DNA? So what do we have? Huh?
Hybrid?
Right, so we have what's functionally N14 and a half. But we can't have a half a proton, can we? Right, so what's the explanation for this? Well, the explanation for this is that in the double helix, right? Well, this was after the Watson-Crick paper was published. So we know that there's a double helix. Well, what if one helix was made up of entirely N15 nitrogens and the other double helix was made up of entirely N14 nitrogens? Well, now we have something that's not all the way N15 and not all the way N14. In fact, it should be exactly in the middle, as we see here. Right, so we should be at this position here. So this is what was expected if this semi-conservative model was what was being done here. And in fact, that's what is observed. So what would happen now at the second generation, right? So now there's evidence that yes, the semi-conservative model is correct. So let's go one further generation, just like we saw here, right? In this case, we have 2 and 2, right? So we have 50% and 50%. 50% should be hybrid, so at which line is this going to be running? In the middle, and we have 50% new, so at which line should this be running? Right, and 14. So is this the case? Well, here we see we